This is the fourth and last video for chapter 12, and as mentioned, we're just going to be going through a few other pathway examples. So the first two of these are bacterial motion and plant ripening, and they are not going to fit neatly into one of our 6 categories because those are human signaling pathways. And then we're going to talk about the senses, human senses, sight, smell, and taste, which do. So bacterial motion occurs generally going in response to food. So that's the main thing bacteria want, is where's the food so we can grow and reproduce. And the way this works, at least in these bacteria are pretty varied, so I don't want to, this is for E. coli. I don't want to imply that they're all exactly the same, is they have flagella, and when the flagella are all moving in a counterclockwise motion, that's going to cause it to run, which means move consistently in one direction. And if they move in a clockwise rotation, they all sort of cancel each other out, and they sort of tumble around in a haphazard way. So the way this works is as if they're running and they're detecting that the attractant is getting more concentrated, they tend to keep running in that direction longer. If they detect that it is getting less concentrated, they tend to run in that direction for a shorter amount of time. And regardless, then they'll stop every so often and tumble and then just head off in some random direction, but sort of eventually getting closer and closer until they reach the food. So the way this works is through basically it's an RTK. It's a slightly different one that's all one component, but it's still a receptor that is a kinase. Well, it's not really an RTK because it phosphorylates not a tyrosine. But it's a receptor kinase, let's call it that. So the attractant here binds the outside. That causes the inside portion to get phosphorylated on a histidine. We've mostly seen tyrosines, serines, and threonines get phosphorylated, but histidines, aspartates, and glutamates can as well. So this gets phosphorylated on the histidine. That causes it to be activated. This might imply that this whole thing is a histidine. This is just a protein domain. The histidine is just the part that gets phosphorylated. That activates it, and then it is a kinase that puts a phosphate onto this response regulator. Again, this is not just an aspartate. That's just the residue that's going to get phosphorylated. That activates this, which then goes over and controls the flagellum. So as long as the attractant is getting more concentrated, this pathway is getting turned on more and more, and we lead to more counterclockwise rotation. If it starts running in the wrong direction, the attractant will fall off because the concentration gets lower. That will slow down this pathway, the response regulator will fall off, and the flagellum will start rotating in the counterclockwise direction, which will cause it to tumble. So that's how basically these completely non-conscious bacteria can still figure out where the food is. All right, the second one is plant ripening, which happens in response to ethylene. Ethylene is just the simplest of the alkenes, two carbons with a double bond between them. This is a gas at room temperature, and this binds to a 2-component system that looks kind of like either the RTKs or the RGCs, but is neither a kinase nor a guanylyl cyclase, so is of course neither of those things. But it's still one of these things where you have two things come together when they bind the ethylene molecule, and that activates this component down here. So what this does is this is going to inactivate CTR1. So CTR1 is a MAP-KKK that leads to a MAP cascade that inactivates IN2. So by inactivating CTR1, we are going to activate IN2. IN2 is then going to act on a transcription factor that creates some mRNA that can also then act on another one. So the mRNA makes this ERF1 transcription factor, and then we make more proteins that are these ethylene response proteins. And these are just things that do important tools in plant ripening. So things that might break down the really hard polysaccharides into shorter monosaccharides, is what makes the fruit feel sort of softer and riper. So ethylene gas is given off also by ripening fruit. So it's sort of a cyclic thing where the fruit starts ripening and then it will give off ethylene gas and it causes all the other fruit around it to ripen. This is the reason that people sometimes say if you want to ripen fruit really fast, you put it in a paper bag, especially if you put it in a paper bag with some already pretty ripe fruit, like a ripe banana. It will ripen faster, and that's because the paper bag will catch some of the ethylene gas. A lot of it's just going to pass right on through, so it's not quite as helpful as I think some people think it is, but that's the idea behind this. All right, so moving on to human senses. We're going to start with sight and spend most of the time on sight. And so just a little bit of the basic biology of the eye. Honestly, those of you who've taken anatomy probably know more about this than I do, but just the basics we need to know here is this is your eyeball looking forward. And so we've got, this is the lens that you see light through. And at the back of your eyeball, we have the retina. And the retina is attached to the optic nerve, which eventually goes to your brain. And that's how we see things. So in this retina, we have a variety of things. We have rods and cones are going to be the most important part of them. And these are all connected to the optic nerve. And when a photon comes in and hits these things in the back of your eye, then we perceive this as different colors of light or just as light, depending on what it's hitting. So the eye has a lot more rods than it does cones. It has about 10 to the 9th rods and only about 10 to the 6th cones. So it has about 1,000 times more rods than cones. Rods are the things that allow us to see basically just black or white, just light or not. So it doesn't detect whether or not there's colored light. It's just, is this light or not? But because we have so many more of them, it is actually more perceptive. So this is what helps us to see in very low light scenarios, which is why you can't really see colors very well in low light scenarios. The cones are what help us determine what color light we are seeing. And there's not as many of them, so that's why they don't work as well when we have very little light hitting our eye, because it tends to not really hit the cones as often. So the receptor that is used in the sight pathway is called rhodopsin. Rhodopsin is a very specific version of a class of molecules called opsins that are all used in sight, and it's a GPCR. So we know that means it has these 7 transmembrane helices. It has a G protein bound here that has 3 subunits. This G protein used in sight actually has a specific name, transducin, but it is exactly the same as the other G proteins that we have seen. The unusual thing about rhodopsin is that it does not bind and unbind to a ligand. It actually has a ligand bound all the time, and this is called a chromophore, and what happens is when the light hits this cis-retinal ligand, it converts it from cis-retinal to trans-retinal. So we talked about this. This is post-vitamin A. So vitamin A is converted into retinal, and it starts out in the cis configuration, and it just converts it to trans when the light hits it. That's right. It takes energy to do that. The light can do that. So really what the rhodopsin is measuring is not whether or not something is bound, but actually whether or not trans-retinal is bound. So when cis-retinal is bound, it does not considered activated, but when trans-retinal is bound, then it's activated. And then the whole GPCR thing where this moves over and goes to an effector enzyme and whatnot gets started. All right, so then the signal that goes to the brain is started by a decrease in cGMP. So we need a start, right? We know that the rest of the pathway is going to involve an action potential with sodium channels, depolarizing the membrane, and all that sort of thing. So what's going to start it out is going to be cGMP decreasing. So here this is very high. This is when it's dark. So when you have not seen anything, so your eyes are closed, or even this is a cell that just hasn't had a photon hit it in a while. So that we're going to have a large amount of cGMP in the nerve cell here. And when the cGMP gets lowered, it causes, this is a ligand-gated ion channel. So when cGMP is bound, it keeps it open, and this is the dark sort of normal way. But when the concentration drops, it falls off, and then the ion channel closes. So the ion channel closing is going to it's going to hyperpolarize the membrane. So beforehand, it's negative 45 millivolts. That's about what we've talked about as being a normal potential. But afterwards, it's negative 75, and that's because we aren't bringing in sodium and potassium are not going to be able to come in. So that's going to hyperpolarize it. That's going to start an action potential that leads us to a signal in the brain. So here's just the entire pathway so you can see how this is all connected. So it starts over here. So light is going to hit the rhodopsin, and this is sort of showing, it's very small, but if you look at the figure from your book, in the book, you should be able to see this a little bit better. This is just showing you the cis rhodopsin the cis retinal versus the trans retinal. And without really being able to see, you can see, right, it's a different shape. So it converts to trans retinal. That causes the alpha subunit here of transducin to dissociate, swap out its GDP for GTP, and go over to the effector enzyme, which in this case is a phosphodiesterase. So it is going to cleave cGMP into regular GMP. In this case, it actually does this by, there's another protein that is going to be bound and sort of inhibiting PDE in the dark, and it's going to move over and bind to the GTP. So that's what's going in here, to the G protein, which is happening here. So in any case, it's going to activate this this phosphodiesterase, which takes the cyclic cGMP, cleaves it, and that lowers the concentration of cGMP, which will close the sodium and calcium channel, which causes lower sodium and calcium inside the cell, which starts the action potential. However, this also has to be stopped relatively quickly because otherwise you would not be able to use the same cell to see anything else. And you all are relatively familiar with what happens if something like that happens. It's when you sort of see spots or something, like you look at the sun and you kind of burn out your cells for a little bit. That would not be good to happen all the time. So basically we want this to turn off relatively quickly. Pretty much as soon as it starts the thing, we want to stop it again. All right, so what's going to happen there, the way that works is, let's start over here. So one thing is that this trans rhodopsin is going to, let's go here. The trans rhodopsin is going to get swapped out for cis rhodopsin. And this is done by the joint actions of proteins called recoverin and arrestin. And the other thing we need to do is we need to remake some cGMP so that we bring that cGMP concentration back up, right? So there is a guanylyl cyclase in here. This is not a receptor. It's not a receptor guanylyl cyclase. It's just a guanylyl cyclase that is going to remake cGMP. And this is also affected by the calcium concentration. So lower calcium concentration, which is what is happening right when we started the pathway. Lower calcium concentration is going to activate this guanylyl cyclase to turn the pathway off. So what the immediate effect of turning the pathway on is also going to turn the pathway off. Up here with the G protein, this is turned off in the same way others are, where this is also a GTPase. So after a pretty short amount of time, it will cleave the GTP to GDP, which then causes it to slide back around to the rhodopsin. I guess what I should have mentioned is what causes this stuff to happen is also that you don't have the alpha subunit anymore. All right, so pretty much all the things are going to reverse itself, and this particular part where the calcium upregulates the guanylyl cyclase, I think is, kind of helpful in understanding how that works. All right, so that is how the rod cells work, and again, like I said, these are just going to detect any photon of light. So any photon of visible light that hits your rod cells is just going to say, hey, I see light. All right. Now, if we want to see color, we're going to use the cone cells. And cone cells, for most people, come in 3 different types. They come in blue, green, and red, and these have different opsins in them. So rhodopsin is the one that's found in the rod cells. You kind of remember it because it kind of looks like rod. I mean, it's got an H, but, you know, rhodopsin. The other opsins are found in these guys, and these are just slightly different molecules, and therefore only detect when lights of the right colors are going to hit them. Okay, so if you have a cone cell that has a blue opsin in it, and you have a 650 nanometer light that hits it, you're just not going to pick it up. All right, and so presumably when you see something that is blue, more than one photon is going to hit your eyes. So some of them will hit the right blue cone cells and you'll see the blue, right? And if it happens to hit a red cone cell, well, that's fine. You just won't see anything there. So red and green are, so blue's kind of over here by itself. Red and green are over there and they kind of overlap quite a bit. And this is the reason that people can be red-green colorblind, because if they are missing one of them, you kind of just, the other one just kind of takes over, right? So if you have, let's say you're missing this green one, but a light right here at this wavelength hits a red cell, that's going to pick up most of that. It's going to pick it up pretty well, right? So you're probably going to see that as red. Now, if somebody who has a normal green cone cell as well sees that light, there will be a lot more that shows green, and that will overwhelm the bits that might show into the red. So, but if you're missing it entirely, the only thing that will pick it up is the red, and that's why red-green color blindness is the most common type of color blindness. Because if you're missing either of these two, the other, you'll just sort of see them both as a very similar color. There are some people that are known now to actually have 4 different colors of cone cells. You might think that the 4th one would maybe be over here near the blue, making a little bit more differentiation there, but it's actually generally in the red-green area. There's another cone cell, and people that have these can detect more subtle differences in colors than those of us who just have the normal 3. Hypothetically, you could also be lacking the blue cone cell. If that happened, you would have much more, you would have a very, very different type of color blindness, right? You wouldn't really be able to see any blue at all. You wouldn't see it as green or red. You just literally would not see it. But I don't think that's as common. It's much more common to be missing one of these two. So interesting side note here, John Dalton of the atomic model fame, 1800s sort of, he was colorblind. And at the time, they didn't know why people were colorblind. And he thought this was really interesting. And one of the things that they thought was maybe some people had slightly colored fluid in their eyes. And so, you know, if you're always kind of looking through green fluid, you just can't really see green, or you can't, something like that, right? So, actually, when he died, he left I'm not sure if he left his whole body to science, but he explicitly told them to dissect his eyes and find out if he was right. So they dissected his eyes. They, of course, found that there was nothing abnormal about the fluid in his eyes. And that might have been the end of it, except for somebody just sort of put them away. They didn't throw them away. They just kind of put them on a shelf somewhere, or in a fridge, or, you know, storage somewhere. Probably actually in a shelf, sort of in formaldehyde or something like that. And they sat there. And then once in the 20th century, once we had figured out the genetics and figured out exactly what was going on, they were able to sequence the genome of his eyeball cells and determined that, in fact, he was missing one of the genes for the opsin. He was actually missing the green one. So you can be red-green colorblind missing either. He happened to not have the gene for the green opsin. This was shown in like the 1990s. So quite a long time later, just because somebody put them away, which is pretty cool that we could find out that's why, you know, Took a lot longer than he anticipated, but we do now know why he was colorblind. All right, so then we're just going to talk a little bit about taste and smell, both of which use GPCRs, or rather I should say some tastes. So generally considered to have 5 tastes: sour, sweet, salty, bitter, and umami, which is sort of like savory. And 3 of these, sweet, bitter, and umami, use GPCR receptors. The other 2 actually just use ion channels, because sour is just tasting acids, so it's just an acid. It's a channel that lets H+ through, and salty just lets in sodium. So those 2 just are ion channels, but the other 3 are GPCRs. So this is going to talk about sweet tasting. So on your taste cells, which are of course on your tongue, you're going to have each taste cell is going to be one particular type of taste. So whether it can taste sweet, sour, etc. So if you're a sweet taste cell, you're going to be doing this one. This G protein also has a fancy name, gusducin. But again, it's just a GPCR. It's just a G protein bound to this. So the sweet receptor. And again, it's going to do the same thing where it goes over. This is just like in the epinephrine pathway, an adenylyl cyclase. And so it also uses CAMP. You might now be wondering, why does it not cause the same effects as adrenaline? And it's simply because different things are active in your taste cells. So CAMP being activated in your taste cells does not lead to the same upregulation of genes that it would in other parts of your body. Instead, it leads to a signal to your brain that says, hey, we taste sweet. And the way that happens is, again, CAMP, remember, activates pKa. And in the taste cells, one of the things pKa can do is phosphorylate a potassium channel. And so this potassium channel, when phosphorylated, is going to close, and that's going to depolarize the cell and start that action potential, telling the brain, hey, I tasted something sweet. So pretty cool. Bitter and umami taste in very similar way, or work in very similar ways. They just have a slightly different receptor. Smell also uses CAMP. Humans have a pretty great sense of smell. I mean, maybe not compared to some other animals, but compared to our other senses, we are able to distinguish many, many different smells. And that's because we have a lot of very sensitive GPCRs that can detect this. And so the odorant is just going to bind to this GPCR. Go over here. It's also going to activate an adenylyl cyclase, causing CAMP to increase. And it's going to, in this case, actually open some CAMP-gated ion channels. It doesn't work through the pKa pathway. That is just going to, again, eventually, so it's a 2-step. So the calcium comes in, and then the calcium opens up the chloride ion channels, which causes chloride to go out, which depolarizes the cell and starts the pathway to, again, the brain. This one's a little bit trickier because odorants are not liquid, or they're not aqueous. So it's a little harder to bind a GPCR if you are in the air. So the way this works is there's a mucus layer in your nose, and that's one reason we have thin layers of mucus, right? Not when you're sick and you have a lot, but the mucus layer actually gets the odorant the odorant goes into the mucus layer, at which point now it can bind to the receptor. All right, so this is just a comparison so that you can really see how similar all these GPCR pathways are. All right, we didn't actually explicitly talk about vasopressin, but epinephrine, right, here we have the beta-adrenergic receptor, our G protein, our adenylyl cyclase, CAMP, pKa, right? We have here as our site, where light hits rhodopsin. This one's this is probably the most different, right? Because we have this different one, and then we have the decrease of cGMP. And then we have our odorants and our sweet taste. Odorants can go in one of a couple different pathways. That's what they're trying to show you. But the odorants and the sweet tastants. And really the one thing you can really tell, this is why this is all one big class, right? They all have the same, generally same three components. What this guy is may vary, but actually we see a lot of adenylyl cyclases. And then they all lead to second messengers. So GPCRs are always going to have second messengers, and then they're going to lead to downstream effects. All right, so that is the end of what we are going to cover for Chapter 12.